Sexual reproduction in plants is a biological process by which plants produce offspring through the fusion of male and female gametes. This leads to the formation of seeds, which can grow into new plants. Fertilization is the process by which male and female gametes unite to form a zygote. There are two main types of fertilization: allogamy and autogamy. Allogamy, also known as cross-pollination, occurs when the gametes come from different individuals. Autogamy, or self-pollination, happens when the gametes come from the same individual. External fertilization is a mode of reproduction in which a plant's gametes are released into the environment, where fertilization occurs outside the organism's body. A common example of this is seen in many aquatic plants. Internal fertilization is a reproductive process in which fertilization occurs inside the organism's body. This method is seen in many flowering plants where pollen is transferred to the ovule within the flower. Alternation of generations is a reproductive cycle of certain plants, fungi, and protists. It involves alternating between two distinct forms, a haploid gametophyte and a diploid sporophyte. The gametophyte produces gametes, sperm and eggs, through mitosis, which fuse during fertilization to form a diploid zygote. This zygote grows into the sporophyte, which produces haploid spores through meiosis. These spores develop into new gametophytes, completing the cycle. This process allows for genetic diversity and adaptation to changing environments. The alternation of generation is significant for three main reasons. First, it allows for genetic diversity through sexual reproduction. Second, it enables plants to adapt to varying environmental conditions. And third, it helps in the dispersal of species, ensuring their survival and proliferation. The structure of a flowering plant is quite fascinating. It includes roots that anchor the plant and absorb nutrients, stems that support the plant and transport fluids, leaves that carry out photosynthesis, and of course, the flowers, which are the reproductive organs. The flowering parts of a plant include the petals, which are often brightly colored to attract pollinators, the sepals, which protect the flower bud before it opens, the stamens, which are the male reproductive organs consisting of anthers and filaments, and the pistil, which is the female reproductive organ consisting of the stigma, style, and ovary. The ovary contains ovules, which develop into seeds after fertilization. Pollination is the transfer of pollen from the male anther of a flower to the female stigma. It's a crucial step in the reproductive cycle of plants. There are two main types of pollination: self-pollination and cross-pollination. Self-pollination occurs when pollen from the anther is transferred to the stigma of the same flower or another flower on the same plant. This method ensures reproduction even in the absence of pollinators. Cross-pollination, on the other hand, involves the transfer of pollen from the anther of one flower to the stigma of a flower on a different plant. This method promotes genetic diversity, which can lead to healthier and more resilient plant populations. Fertilization in plants is a complex process that begins when pollen grains land on the stigma of a flower. The pollen grain germinates, forming a pollen tube that grows down the style towards the ovary. The pollen tube enters the ovule through the micropyle, a small opening that facilitates the entry of the male gamete. Inside the ovule, the male gamete travels through the pollen tube to reach the egg synergid cells, which help guide the sperm to the egg cell. The polar nuclei in the central cell fuse with another sperm cell to form the endosperm, which nourishes the developing embryo. The antipodals, though not directly involved in fertilization, play a supportive role in the development of the seed. Fertilization occurs when the male gamete fuses with the female gamete, leading to the formation of a zygote. This zygote then develops into an embryo, marking the beginning of a new plant life. Double fertilization is a complex process in flowering plants where two sperm cells from a pollen grain fertilize cells in the ovule. One sperm cell fuses with the egg cell to form a zygote, while the other fuses with two polar nuclei to form the endosperm, which nourishes the developing embryo. This process ensures that the seed has a food supply for germination. Seed formation is a crucial process in the life cycle of plants. It begins with fertilization, where the male gamete fuses with the female gamete to form a zygote. This zygote then develops into a seed, which contains the embryo of the new plant. The seed is protected by a seed coat and contains stored food to nourish the embryo during germination. Seed formation ensures the continuation of plant species and allows for the dispersal of plants to new locations. The structure of the micropyle is quite fascinating. It is a small opening in the ovule of a seed plant. Through this tiny pore, the pollen tube usually enters the ovule. The micropyle also allows water to enter the seed. This is crucial for the seed's germination process. The micropyle plays a vital role in the development of the new plant. Understanding its structure helps us appreciate the complexity of plant reproduction. The endosperm is another critical component of seeds. It serves as a food reserve for the developing plant embryo. For example, in grains like wheat and corn, the endosperm is a significant source of nutrients. Cotyledons are the first leaves that appear on a seedling. They are part of the embryo within the seed of a plant. The radicle is the part of a plant embryo that develops into the primary root. It is the first part of the seedling to emerge from the seed during the process of germination. There are several key differences between monocotyledonous and dicotyledonous plants. First, monocots have one cotyledon, while dicots have two. Second, the leaf veins in monocots are usually parallel, whereas in dicots they form a net-like pattern. Third, the vascular bundles in monocots are scattered, while in dicots they are arranged in a ring. Fourth, monocot roots are typically fibrous, whereas dicots often have a main taproot. Fifth, the flower parts of monocots are usually in multiples of 3, while in dicots they are in multiples of 4 or 5. Lastly, monocots generally have pollen with 1 pore or furrow, whereas dicots have pollen with 3 pores or furrows. Fruit development is a crucial stage in the life cycle of flowering plants. It involves the transformation of the ovary into a fruit, which protects the seeds and aids in their dispersal. This process is often triggered by fertilization and involves various physiological and biochemical changes within the plant. Now moving on to the methods of seed dispersal. Seeds can be dispersed by wind, by animals, or by water. There are 5 adaptations in the structure of insect-pollinated and wind-pollinated flowers. Insect-pollinated flowers often have bright colors, sweet scents, and nectar to attract insects. They also have sticky or spiky pollen to cling to the insects. Wind-pollinated flowers, on the other hand, typically lack bright colors and scents and produce large quantities of light, smooth pollen that can be carried by the wind. Their stigmas are often large and feathery to catch the pollen from the air. Germination is essentially the process by which a seed develops into a new plant. There are 5 key comparisons between epigeal and hypogeal germination. In epigeal germination, the cotyledons are pushed above the ground, while in hypogeal germination, they remain below the soil. For example, beans and sunflowers exhibit epigeal germination. Epigeal germination typically involves the elongation of the hypocotyl, whereas hypogeal germination involves the elongation of the epicotyl. Peas and maize are examples of plants that undergo hypogeal germination. Epigeal germination is common in beans and sunflowers, while hypogeal germination is found in peas and maize. In epigeal germination, the cotyledons become photosynthetic, but in hypogeal germination, they do not. Finally, epigeal germination usually results in a quicker initial growth compared to hypogeal germination. The conditions necessary for the germination of a seed include water, oxygen, and the right temperature. Water is essential to activate the enzymes that begin the growth process. Oxygen is needed for respiration, which provides the energy for growth. And of course, the temperature must be suitable for the enzymes to function effectively. What is an embryo? An embryo is the young multicellular organism before it emerges from the seed. In plants, the embryo develops after fertilization and is enclosed within the seed. There are 2 types of seeds, endospermic and non-endospermic. Endospermic seeds retain part of the endosperm as food. Non-endospermic seeds store food in the cotyledons.